We are finishing up our discussion about the innate system. We talked about the first line of defense is basically our surface barriers, and now we're dealing with all of the other things that are considered the second line of defense for us, and we'll get to our immune system as actually the third line of defense. So all of these cellular as well as chemical components here are considered in that broad category of second line of defense that is offered to us by the innate system. I want to take a look here at this. I've rearranged the notes, but it's not with your notes right now. Let's go ahead first and look at this picture right here and realize that this discussion is over complement, and complement's 30 different plasma proteins that are in the bloodstream circulating all the time in an inactive form. And certain numbers will become activated by various things. And whenever the certain ones get activated, they can do certain things. So complement does a lot of different things, not just one thing. And one of the things that complement can do is this opsonization process right here. And the little C right here is our complement. It is attaching to a bacterium. The bacterium's got these little blue bubbles on it, and those are basically just polysaccharides. Those are little sugar complexes. But let's just realize that this complement gets stuck to this bacterium, and by the sheer fact of this type of complement sticking to this bacterium, it makes this macrophage notice the bacterium even better and makes the macrophage basically engulf the pathogen for us more efficiently. So that's opsonization. When the complement does that, when complement attaches to a bacterium, for example, and makes it more attractive for macrophages, it is called an opsin or opsonin. So that's one of the things to realize that complement can do for us. It can attach itself to a bacteria to make it more attractive to a macrophage. There's other things that actually make a pathogen more attracted to macrophages, and that's why it actually has a special name called an opsin or opsonin. Complement also, some of it, when it gets activated, can also help to increase inflammation. And remember, inflammation is designed to wall things off so that the pathogen is not able to spread. And so with inflammation, we get the mast cells and the basophils, which degranulate to actually help with the leaky capillary situation. And then the neutrophils and macrophages will also be called in to actually then help fight that pathogen. So some complement can actually do that. Not all complement does. Not each member of the complement will be an opsonin or inflammatory agent or the cytolysis stuff. So the cytolysis is related to that MAC that we talked about at the during our last lecture. MAC stands for membrane attack complex, and there are two ways that complement can cause MAC formation on the cell, and that's cytolysis. But whenever MAC does occur, whether it's antibody mediated, which is the classic process, or whether it's the polysaccharides, which is the alternate process. And right now this picture is showing us the alternate process right here. So it's the polysaccharides on the bacterium which make it attractive and activate a specific complement component that will create MAC or membrane attack complex. And this will actually bore a hole into the bacterium and cause water to flow into the bacterium, and then we actually have that lysis. And so that's an important aspect that complement can do. So interferon is for viruses, and complement are for actually cellular things such as bacteria and even transplanted cells or blood, for example. Fever. So this is also an innate mechanism. Fever is defined as an abnormal elevation of body temperature, and the chemical that causes this will be called a pyrogen. So pyro is fire, and pyrogen is going to be a chemical that will help to reset the hypothalamus, because the hypothalamus is that area of our brain that sets what our body temperature should be. It will reset the hypothalamus to a higher temperature. And this higher temperature is going to actually affect zinc and iron levels. So Zn is for zinc and Fe is for iron. So this pyrogen, an elevated temperature, will affect zinc and iron levels that are in the bloodstream. It will end up being sequestered in the spleen. Sequestered means that we're going to preferentially take it up and remove it actually from the bloodstream, so effectively dropping circulating zinc and iron levels in the bloodstream. The important thing about this is that zinc and iron are needed for bacterial replication. So this effectively slows down the bacterial division and replication process for us. Things that are pyrogens are various toxins, released by bacteria, can act as pyrogens. Also, our WBCs and macrophages can actually release chemicals that serve as pyrogens. So WBCs and macrophages. So this elevated body temperature helps us to decrease the replication of bacteria, decrease the replication of viruses. It actually helps to increase interferon activity, increases the activity of the actual adaptive immune system, and also increased body temperature will increase tissue repair. So that's the benefits that are actually observed in all of these situations with increased temperature. So a little bit is good and a lot is bad, because remember, excessive body temperature can actually start to denature proteins. Now we're going to get into the adaptive branch, which is more effectively called immunity, and it is considered our last line of defense, or the third and therefore last line of defense. And it is very specific because it is a system that has recognition. The other system just kind of says, well, you fit into the category of a virus or you fit into the category of bacteria. This is a very specific recognition system and very particular as to what it actually is recognizing. The characteristics of this recognition aspect is antigen specific. So antigen is going to be abbreviated AG for antigen. And recognition is specific, and we'll get into the specific aspect in just a moment. Also with the immune system, it is systemic. What that means is, remember, with our innate system, we are trying to actually localize the infection or the pathogen, but that once that pathogen actually breaches our attempts to keep it localized. It then circulates around into the system, and that's really bad for us. But because our immune system is systemic, it doesn't matter where that pathogen is. We can get to it no matter its location. Also, our immune system, because it has a recognition capacity, it has memory capacity. And that's another reason why we like to do immunizations or vaccines, because of this very important part. Memory, it remembers prior infections and will respond in a much more rapid fashion if it then encounters the real thing, such as after a vaccine. Two main branches of our immune system are the humoral immunity or cell-mediated immunity. Humoral immunity is from our B cells. Cell-mediated immunity comes from our T cells. And again, humoral immunity is antibody related, so AB is going to be our abbreviation for antibody. So antibody mediated. Protection is by proteins in the plasma. And so it's humoral. Cell-mediated immunity, protection is by cells. And what this means is that these cells are actually going to physically interact with other cells. So physically coming into contact with other cells. Let's look at this flow chart, and we'll talk about a few things. And as we go through our material, you should be able to refer back to this flow chart and have a slightly better understanding. So adaptive immunity, cell-mediated, T lymphocytes. Humoral immunity, B lymphocytes. In the notes, it does say what they're effective against. And as we go through our notes here, come back to this and see where in our notes we actually discuss the antigen outside the cells or the antigen within the cells. Outside the cells, it's an interesting aspect because the B lymphocytes don't actually have to come into contact with the cell once antibodies are actually being, the foreign material, once the antibodies are actually being made against it. Of the T lymphocytes, there are two big players, the cytotoxic T lymphocyte and the helper T lymphocyte. We will talk about cytotoxic T cells as T little c, and we will talk about helper cells as T lower h. The cytotoxic T cell is truly the destroyer, and it destroys the cell by apoptosis. And apoptosis is going to be our perforin and granzyme process. Antibodies are going to be what are the final product from our humoral immunity, and antibodies are going to do different things, and we'll discuss that. But the cells that actually make antibodies are actually called plasma cells, which is, again, humoral immunity. So a B cell will be transformed into a plasma cell. So that means the plasma cell is actually actively creating antibodies that it's injecting into the bloodstream. Let's talk about antigen, AG, a substance that can turn on the immune system. These are actually quite large molecules, normally not present in the body. In order for an antigen to work effectively for us to create a response, it has two properties here called immunogenicity and reactivity. And as we go through the notes, we'll understand immunogenicity and reactivity a little bit more. Immunogenicity, in our notes state, can be recognized by the immune system as foreign. And our problem is some molecules are too small to be recognized. So, and above it said large molecules. So we're saying that these molecules are actually too small for the immune system to actually recognize. So immunogenicity is a recognition process. So keep that in mind, recognize or interact. The other thing too is some of our molecules, cellular molecules, and I'll say markings, are actually similar to cellular molecules of antigens. Let's say pathogens and then antigens. And when we get further on into imbalances, that's one of the problems with rheumatic fever. The Streptococcus, for some people, actually has a surface marker or surface protein that's very similar to a surface marker or surface protein on heart valves for them. And so we are actually designed not to recognize the surface marker on our heart valves, and so when we get those people get that type of Streptococcus infection, they can't recognize it because their immune system has trained them to recognize self, and this Streptococcus has something that's just like their own self. So that is inability to actually be recognized, immunogenicity. Reactivity, the ability to actually turn on or activate immune cells. So it's really interesting. It's a two-step process. We have to recognize and then we have to turn on our immune cells in order for us to get a response for that. And this is a sequential activation process. And we are going to learn it requires T helper cells for this. This is a very T helper cell dependent process, reactivity. Hapten is called a complete antigen because it's the one that's too small to be recognized. So it's smaller than 10,000 molecular weight. So things over 10,000 molecular weight are large enough to be recognized or have immunogenicity. And because it's too small, it lacks immunogenicity. Or ability for our system to recognize. What's interesting, haptens can attach themselves, not all of them, but some haptens can attach themselves to proteins within our system. And one of the proteins that they can easily attach to within our system is albumin. Albumin is from ourself, but it's big. And suddenly, if a hapten is able to actually attach to albumin, it's like suddenly making itself a much bigger presentable molecule. And then that is something that we can actually recognize and attack those haptens. Now, this is a problem that causes allergies or allergic reaction. And we'll talk about penicillin for a moment. This is typically what actually brings about penicillin allergies. Now, when people take penicillin, do they take it for a bacterial or a viral infection? Bacterial, because it is an antibiotic that is specific for bacteria. So if you have a viral infection, will it help you at all? No. So a lot of problems are people really want to feel better when they're sick, just like right now I want to feel better when I'm sick, but you know, we work on denial and distraction. Works actually sometimes quite well for us. But when that's not working and you go to the doctor and the doctor says, well, you know, it's going to be about a week before you're better, and you says, well, can't you give me some antibiotics so I can get better faster? And that's what antibiotics will do. If you have a bacterial infection, they'll help you to get well and much faster. But if it's a viral infection, it won't help you to get any better other than what it can do via the placebo effect. And do you guys know about the placebo effect? A lot of people do. Just by taking a pill, you feel better. And that's called the placebo effect. And that's why drug studies always have a placebo control. Because when you give somebody a medication, the medication should be better than placebo. And so what that means is that placebo has typically at least a 20% improvement rate for most people, and sometimes even more than that. So medications are not viewed as effective unless they are actually better than placebo, and you would actually really want them to be significantly better than placebo. But that's not always the case. Sometimes they get approved if they're just a little bit better than placebo, not significantly so. But back to penicillin. So people want penicillin. They go to the doctor, I'm sick, I want an antibiotic, and so the doctor gives you an antibiotic, or a healthcare provider gives you an antibiotic, and you don't have the bacteria that penicillin is designed for. So that means when you take penicillin, it's circulating around in your system for a long time because it's not being taken out, because it's not finding the offending agent that it was designed to actually fight. So it's circulating around in your system for a long time, and it has a lovely tendency to actually bind to albumin when it's hanging out for a long time. When it binds to albumin, it is no longer a material that's going to be removed from your system rapidly. It is now actually was a hapten and now is recognizable, and now it's immunogenic, and you can actually mount an immune response, and unfortunately you mount an adverse immune response to this. And so the first time this happens, you don't really have any symptoms, but the next time, because the immune system has memory, you take penicillin, and it might even be needed for an actual bacterial infection. But because you have now primed your system to actually call penicillin an allergen, you will have a rather rapid response to it. And so that's why taking antibiotics for a viral infection is kind of stupid. And also it helps to create a problem of drug resistance, not just allergies, but drug resistance. And so, but our society is pretty pill-oriented, isn't it? Don't pills make you better? So it's really hard to get people to be okay with not being given a pill. And so if people aren't happy with that one doctor or caregiver not giving them a pill, they'll go find another one that will very often, which is unfortunate. But that's the hapten story. Too small to be recognized, but can unfortunately become recognized if it attaches to a larger molecule. Antigenic determinants. The precise location on the antigen where antibodies physically attach. I want you to realize antigen and antibodies seem to be already linked in our discussions here. So an antigen, and so you have to memorize and keep it straight in your mind what an antigen is and what an antibody is. An antibody sounds like antibiotics, which are good for you typically. So antibody fights the bad stuff. The antigen, remember, is foreign material, a large molecule is foreign material not normally present in your body. So antigen is AG, antibodies is AB. And antibodies fight antigens. So that's a very simplistic way of looking at it, but it's a very effective way to actually look at it. But an antigen is a large molecule, typically. Like a bacteria is a large molecule. It's more than just a molecule. It has a whole bunch of different molecules. And so what we're trying to tell you here is that on an antigen, such as a bacteria, there are many different areas that are the hot spots, so to speak. And we call those hot spots, which we will be able to recognize and react to, as antigenic determinants. So on a pathogen, and I'm putting in parentheses antigen, there are several different areas we can react or that are immunogenic to. And they are called antigenic determinants. For example, I'm just going to draw a long bacterium here, E. coli or something along that line, and I'm going to put some circles on them, and I'm going to put some squares on them, and some triangles on them. And all of those are meant to show you these are very specific markers, surface markers on this bacterium that we can develop antibodies to. And so if we are a circle, antibodies are going to typically be Y-shaped. So a Y is going to be an antibody. And the important parts of the antibody are these little prongs off the Y. And so this antibody is designed specifically for the circle. Then this antibody is going to be specifically designed to recognize and react to the square. And this antibody is going to be designed to specifically recognize and react to this triangle. So antigenic determinants will have their own specific antibody because they all look different, and the antibody is only going to react to, recognize and react to that thing which is what it was designed to recognize and react to. Here's another one. Some very special antigen types or antigenic determinants we have names for. We have names for those that are associated with red blood cells. And we call those antigens or antigenic determinants agglutinogens because they cause agglutination. Agglutinogens. These are very specific for red blood cells. And what are our red blood cell types that we actually have? We have A, we have B, we have O, and we have AB. And so that's what we're actually talking about. On the surface of our A cells are the A agglutinogens. On the surface of our B cells are the B agglutinogens. On the surface of our O cells are no agglutinogens. And if we are AB, we will have those agglutinogens, A and B. So that's referring to those surface markers that are recognizable, because we have to recognize and then we have to react. Immunogenicity and reactivity. The actual reaction that we see is agglutination. Then another specific antigen type is called an allergen. So it's a surface marker, surface antigen on a cell that will actually initiate an allergic reaction. So we call those agglutinogens, or not agglutinogens, antigens, antigens, allergens. And then we're going to get into a little bit more discussion of the MHC proteins. Because suddenly, previously I told you an antigen was a large molecule not normally present in your body. Now I'm telling you that you have self-antigens. And self-antigens are called MHC proteins, and M for major, H for histocompatibility complex. So MHC proteins are our self-antigens, and that's one of the things that the T cells get trained about. They get trained about the ability to recognize self. And then when non-self comes in, it's easier for them to actually mount an immune response. So MHC proteins, histocompatibility, are something that is tested very stringently with certain type of surgical procedures here in the United States. Do you guys have any idea what surgical procedure we would do? A lot of testing on the patient that's getting the surgery and on the donor that's donating an organ. So MHC proteins are actually what is looked at whenever we do transplants, because the more compatibility you have of one person's MHC proteins to the other MHC proteins, the less likely the rejection. Also, as a agglutinogen, do you want to give a person who's type A blood, type B blood? Because that will cause this immune system type of response. It will actually cause a blood transfusion reaction, which is the agglutination. So you're giving somebody blood, and now it just clumps all out of their blood, which is totally counterproductive, and also puts a lot of large particles in the bloodstream of the individual and can kill the person. So MHC proteins, real important, and we're going to get into the MHC proteins as we actually talk about our T cells and our B cells. So B cells, lymphocytes, because whenever I say a B cell or T cell, remember always they originally were lymphocytes. The B cells, and all lymphocytes come from where? Where do they originate from? The bone marrow. So remember, B cells, lymphocytes, main cell of humoral immunity, they originate and are trained in the bone marrow. And when I say trained, I mean the term immunocompetent. In other words, they're trained to do their job. T cells, they did originally originate in the bone marrow, but they are trained in the thymus. You never find a B cell in the thymus. You only find T cells, but you can find the precursors to our T cells in the bone marrow, because all lymphocytes originate in the bone marrow, but their training for T cells is actually in the thymus. That's where they become immunocompetent. Another big player in the immune system are APCs, antigen-presenting cells. The cells that function as antigen presenters are macrophages, dendritic cells, and even B cells can actually function as APCs, an antigen-presenting cell. what happens is they engulf the pathogen, they digest it, and then they present it to our T cells. And more specifically, the T helper cell is responsible for activation or reactivity. Dendritic cells are kind of the best at this, but macrophages do a good job and B cells do too. Receptor complexes. So we're going to have to be able to have something dock here if we're a receptor. Indeed, they're protein complexes that are on the surface of our lymphocytes that are designed to bind only a specific antigen or antigenic determinant. So when we talk about antigens, we often will drop the word determinant, but I want you to realize if it's a bacterium, for example, it will have many different antigenic determinants. If it's a molecule, it can actually even have, if it's a large molecule, several different areas that are antigenic determinants. So the receptor complexes that we are interested in are our T cell receptors. So T cell receptor complexes on their surface and B cell receptors. So these are protein complexes on their surface designed for a specific antigenic determinant to dock there. Let's take a look at the cartoon right below receptor complexes. And we have T cells on the left, B on the right. The T cells, two main types. We want the T helper cell, and our picture is showing a little tapered cup here as the T cell receptor, but I want you to associate T helper cells with this surface marker, CD4 protein. A CD4 protein is that T cells, actually MHC, to let you know it's a helper cell. So CD4 equals T helper cell. On our cytotoxic T cells, we have a surface marker called the CD8 protein, which identifies a T cell as a cytotoxic T cell. So CD8 is equivalent to cytotoxic T cells, and CD4 is equivalent to T helper cells. They both have their own specific receptors, but these receptors can be for the same type of antigenic determinant. On the B lymphocyte, this is a Y thing, and the Y things are actually a very specific type of antibodies. That's the B cell receptor complex. So these are attached to the surface of the cells for specific binding, because the binding is going to occur right here, and right here, of very specific antigenic determinants. You can always recognize a B cell that has not been activated because it has all these Ys or antibodies on its surface. Once a B cell is activated, those Ys are no longer on its surface because then it's actually working on, because these are for recognition. So these receptor complexes are for recognition. And recognition is synonymous with immunogenicity. And so we have areas right here that we actually have to have docking or binding, and that is the recognition process. And then we can actually work on creating a reaction, which is the two-part step for an antigen to actually have any effect for us. Going back to MHC, major histocompatibility complex, indeed, it is like a cellular fingerprint. That's why we do MHC testing for of the tissues of a kidney donor, and also MHC testing for the recipient. They have to be almost exact in order to minimize our rejection and need for anti-rejection medications. So we call MHC very similar to a cellular fingerprint. There are two classes of MHC that's super important. Class 1 is found on all nucleated cells. So what cell doesn't have class 1 MHC? Red blood cells, absolutely. So it's on red blood cells, we don't have MHC1, but we do have agglutinogens on the surface of red blood cells, which we recognize and react to. So class 1 MHC is found on all nucleated cells, and what's important is I liken the MHC1 receptor area on the surface of a cell, of any cell, of a white blood cell, of a kidney cell, of a liver cell, of a skin cell, like the receptor for a flagpole. So it's designed to hold something there. And it holds and displays an antigen, because now we're talking about antigens no longer being always considered foreign, because our own cells have surface markers. And we'll display an antigen that basically comes from self or foreign. So this is a holder on the surface that the cell will then put in a little flag saying, I belong to the United States of America. I have my little American flag up here. But if it gets invaded by a pathogen, we take out the U.S. flag and we put in the flag of Russia, for example. And so that's what MHC1 is all about. I'm me, I belong here. This is my place. Or I really shouldn't be here, but I am. And so that's why it says displays antigen that indicates self or foreign. And so in order to do this, this surface receptor displays or holds parts of self or parts of the foreign material. And remember, foreign material is actually now antigenic determinants. If you're foreign, that we want to be able to recognize. So it's saying, if the cell is healthy, it's waving its flag saying, I'm happy, I'm me, no problem. But if the cell has turned into a cancer cell, no longer does a cancer cell have everything that's normal about the cell in it, and those abnormal pieces will be actually displayed on the surface so that we can recognize the cell as being as one that's undergone malignant transformation, and we can take it out. Same thing with a virally infected cell. If we are a virally infected cell, we'll be putting things out on the little flagpole waiver so that the immune system can see this immunogenicity, and then once it's turned on, can react to it. And that's what's really important. We're actually all right now just talking about recognition or immunogenicity, and then we'll be talking about reactivity. Class 2 MHC is the next MHC I want to discuss. It is only on APCs. So that means it is on macrophages, dendritic cells, and B cells. Those are the only cells that have MHC2. Do those cells have MHC1? Yes, because all cells except red blood cells have MHC1. So our Macrophages, our dendritic cells, and our B cells have two MHCs. They have the MHC1 and they have the MHC2. And these guys are important because they are actually the ones designed to present the antigen to the CD4 cells. So CD4 cells are so that's synonymous now with TH, T helper cells. So T helper cells don't listen to any other cell except for dendritic cells, macrophages, and B cells. They don't care what other cells say. They care about what these guys say. So T helper cells are going to be very vital in the activation process, because that's actually what they do. T helper cells activate our immune system cells. Activation is a process of cloning and turning on. So what's a clone? It's an exact same cell. So this is a mitotic process of the cells that they turn on where they make a whole bunch of themselves. So that's a cloning process. And in addition to being cloned, they are allowed, they have been given permission to fight, to do what they were trained to do. So activation is very dependent upon T helper cells. And so they will only turn on cloning processes and give directions to actually start fighting if they get the proper information to them. So that's why they won't respond to other cells. They will respond to the cells that actually are, quote, in the know. And when they're in the know, they will then go ahead and T helper cells will actually clone themselves and also be producing chemicals that will actually activate other cells in the system. So MHC2 is very important. So these guys are required to activate TH cells. And that is part of reactivity. So immunogenicity, recognition, and reactivity, activation. That's a T helper cell process. So this cartoon down here is showing CD4 and CD8. Well, CD4 is T helper cells, CD8 is cytotoxic T cells. Let's concentrate just on thinking about CD4 for just a moment. So here is a nucleated cell. This is an MHC with, if we're talking about we want to turn on a T helper cell, are we going to be working on introducing an MHC in class 1 or class 2 to our T helper cell? We want class 2 for T helper cell. We'll talk about this in a moment or next time when we want to talk about cytotoxic T cells. So the nucleated cell is going to bring its foreign material to the T helper cell, and it's going to be loaded up in the MHC2 area. And with the MHC2 area, this will then turn on the T helper cell, and it will clone and it will actually create more chemicals that are required to turn on or activate other cells. So we'll stop there and do a clicker.